Hello, I am Dr. Emily Stein. My title of my talk is The Microbiome and How It Relates to Health and Disease. So a little bit about me. I am a trained microbiologist. My undergraduate was focused in molecular pathogenesis and microbial pathogenesis. I spent about 4 years in a lab at the University of Iowa studying Mycobacterium tuberculosis and its ability to co-opt host immune cells to grow. Really fell in love and became fascinated with the microbiology side of it, and so pursued a PhD at Berkeley in microbial biology, where I spent 6 years understanding the role that microbes play both in response to nutrient stress and starvation, or exposure to infectious or toxic chemicals or heavy metals, and how they really work together to survive that stress in a community. Then I pursued the flip side of things to learn more about host immune function in chronic inflammatory disease states, and spent 7 years as a research fellow in rheumatology at Stanford, focusing on understanding the molecular underpinnings driving neuroendocrine and immune dysfunction in autoimmune disease. Interestingly, there's a microbial component to that as well. And now I am a technical CEO of a small biotech company called Primal Health. So the story of the microbiome honestly begins way back hundreds of years ago by some dude named Antony van Leeuwenhoek. And he was a real outside the box kind of thinker, and came up with this nifty little microscope that when you held it up to the eye pretty close, he could actually visualize single-celled organisms. He called them animalcules, but he wound up being the first one to visualize microbes. And later, around 200 years later, this very brilliant scientist called Robert Koch really started to put 2 and 2 together to understand that, hey, these microbes can actually cause disease. And so through Koch's postulates and all sorts of work that he really did to in both tuberculosis, which is my love, and then malaria and other things, really showed that microbes can be infectious, that they can be transferable, and also vaccine development can occur. And then most recently, there's this wonderful scientist named Jeffrey Gordon. He's at the Washington University in St. Louis, and he's been coined the grandfather of the microbiome. He's done a lot of work more on the molecular side, really showing the benefits or potential harm that can come to a host that has a microbiome constitution that is either a healthful one or a harmful one. And we're going to go into that more later in this talk. So just to make sure we're all coming from the same page, the microbiome is now often coined the forgotten organ of the body. It's a very diverse microbial community. It coats every single surface inside and out. Particularly the GI tract is the most predominant, well-studied microbiome in animals and humans. It comprises fungi, bacteria, viruses, parasites, and they all work together. And sometimes it works well, sometimes it doesn't. It's as unique as your fingerprint. So we all have our own microbiomes. Yes, there are very commonly held species that transfer between human to human. It's, we all have, for instance, E. coli in our guts usually, or some kind of Clostridial strain in our gut. But strain specificity is as unique and individual as a fingerprint. And I'm going to just cover today, since this is a top level introductory talk, talk about the microbiome and introduce you to how it's related to and drives some big things that dictate health and disease, which is development, metabolism, and physiology of animals. So there is a specific kind of language that we microbiologists, particularly in the microbiome sciences, like to talk about, which is dysbiosis. And so just a quick synopsis of that. So the microbiome plus the host, usually in a healthful scenario, are in this positive symbiotic relationship where both sides of the equation benefit. But over time, as animals age, or they've been exposed to various insults, that careful balance can actually be disturbed. And it can either occur in acute phase or over time in a chronic state, which can lead to downstream inflammatory responses or an inflammation of the tissue. Dysbiosis oftentimes in a healthy animal is very rare to see. Most of the time we see animals that have certain kinds of diseases, particularly chronic diseases, tend to have dysbiosis. So that's characterized as a lack of diversity, low abundance of the quote unquote beneficial microbes to inhabit that animal, as opposed to a healthy animal, which usually has a very diverse repertoire of the microbiome and its constituents and abundance of beneficial microbes that really are associated with healthier outcomes. So dysbiosis can actually be caused by antibiotic exposure, chronic stress, limited diet. In fact, a lot of animals that have been shelter animals, given up or have been mistreated and have been rescued tend to have dysbiosis for very many reasons. And the downstream effect of that is usually some kind or multiple chronic diseases. So the first overarching topic I want to just discuss is development. So animal development is been widely understood in its role in the microbiome, and also the microbiome's influence on development has been studied now for over 40 years, mainly in small animals, mainly in academic settings. Things that we do know that might be relevant to your practice are that animals with dysbiotic microbiomes tend to have poor sperm count, poor placenta health. This actually can affect breeding, success rates of breeding. Not only that, but the microbiome has also been shown to actually drive and regulate maternal hormone regulation and cycles, and also fetal neuronal development, particularly has been studied in terms of the gut microbiome and its role in a gut-brain axis and neuronal development of the fetus. Microbiome has also been shown to facilitate nutrient uptake, which is really important for fetal development, but also the maternal diet shapes the microbiome. So it's kind of a chicken and egg scenario where a maternal diet can affect, significantly shift the microbiome, which can then significantly shift nutrient absorption. And so we just need to be mindful of that. And that's a whole other lecture by itself. The microbiome, believe it or not, actually has been shown to shape fetal development in the placenta. Certain biofilms have now been shown to exist inside the placenta, coating the fetuses. And that work is pretty revolutionary. It's still early days, so we'll see exactly what kinds of inner relationship occurs in utero from such an intimate manner. But we do know that the microbiome is an inheritable trait from the maternal microbiome. So even in post-birth development, the offspring inherit the maternal microbiome, and that helps to dictate birth weights, weight gain, certain types of allergies to foods, and carriage rate. So in an animal with dysbiosis, that mom can actually have a risk of preterm birth. Her offspring can have lower birth weight, and her offspring can also be more prone to illness compared to the same kind of an animal that had a healthy, stable microbiome. The really cool thing is that now with fecal transplant technology, we're actually able to show that you can transfer microbiota from a healthy organism to a sick organism to shape the outcome. Other ways that you can also facilitate that is something through diet. So we've known now over the past 20 years that fibers can actually rescue poor diets, rescue poor microbiomes. And it's through these things called small chain fatty acids that are the byproducts of the digested material that these gut microbes make when they are exposed to fibers that are actually beneficial for the host animal and for development. So moving on to metabolism and the role of the microbiome in metabolism, it's fairly obvious. These microbes tend to feed the host. So they eat first, they inhabit the gut. We're talking trillions of them, and they're little energy factories, and all they do all day long is absorb nutrients, break things down, and secrete waste products that then get absorbed by the mucosal epithelium of the host. So not only do they provide a physical and enzymatic process and role in helping host animals digest their foods, but and metabolize their drugs, but they're also foundationally play a role in gut function and neuroendocrine function. So they secrete, now it's been tabulated, over 100 different molecules that stimulate these enteroendocrine cells in the GI tract. And those are the ones that secrete or make the serotonin. So about 90% of an animal's gut actually is responsible for serotonin production, 10% in the central nervous system. And it's through the bacteria and what they're doing that are actually driving hunger cessation and gut feedback loops. In addition to, they're helping drive and tolerize mucosal immune system and regulate the immune responses in the GI tract. And then they're also involved in helping facilitate water retention and gap junction integrity of the intestinal epithelium, which regulates absorption, which then has a downstream effect on muscle gain of that animal. Dysbiosis affects significantly in the gut microbiome. We've seen that dysbiotic microbiomes in the GI tract, those animals tend to have a lot of leaky stool or loose stool, leaky gut syndrome. So they tend to be more inflamed generally. They can have irritable bowel. Some of them tend to be obese. Some of them tend to have diabetes. So insulin regulation is wonky in those animals, and they're unable to actually effectively absorb the right kinds of nutrients. So they're oftentimes malnourished, even though they may be overweight. And this has relevance to your practice because whether you're a large animal veterinarian or a companion animal veterinarian, both the breeding world and the production animal world or the performance animal world is foundationally impacted by the microbiome. Now we're turning a little bit into the physiology. So as I mentioned before, the microbiome is a forgotten organ, and it's an organ that touches every single system of that host animal. And so it's through both physical intimate contact with tissue, or more oftenly identified in research is it's through what they secrete that drives certain metabolic and physiologic processes to occur. So they play a role, for instance, in the mouth, both proximally in the gum tissue and on the tongue and on the teeth, but also distally because they're now linked to cardiovascular and in other and renal systems as well. So they're just like a top level. I mean, to talk about physiology is pretty much every anatomical area of the body and its role in physiology that microbes drive or have play a part of, both in driving and regulating immune tolerance, so innate immune cell activity, but also adaptive immunity. We're finding that memory is also directly linked to gut microbiome. Tissue integrity. So not just the mucosal gut, but also the skin. The integumentary system is driven by who lives in that biofilm on skin as to whether or not hotspots occur. And then neuroendocrine function, obviously sleep-wake cycle, restfulness, aggression, those kinds of things have actually been linked to microbes that inhabit the GI tract. And then feedback loops. So they're intimately involved in not only hormone feedback loops, but also neurotransmitter feedback loops and metabolic feedback loops. And their gene expression actually drives a lot of the host gene expression. And so one of the most foundational things that you can do as a veterinarian is to actually assess quickly how healthy their gut microbiome is, how diverse it is, as a key indicator of whether or not they're going to have some physiologic downstream problem. The relevance to you, for example, are two of the most common reasons why you may see some office visits. Between simple examples are ear infections, and then also dental disease, which is my area of focus. But it's been very well documented that the ear canal contains numerous biofilms of various different types based on location. Same with teeth and same with gums, some gingival locations. There is a different microbiome in every square inch of tissue. And the diversity of that actually directly correlates with the amount of inflammation of that tissue. And so as you can see on the left-hand side of the slide, a clinically affected dog with an ear infection tends to have a ton of fungus and of one species and a ton of bacteria of one species and on the skin as well, compared to that of a healthy dog, which is a very diverse repertoire of microbiome that inhabits that ear and is also found on the skin. The same thing is we're seeing in the mouth of dogs and cats, and we're studying horses as well. And that correlates strongly with our human data, which shows that animals with periodontal or gum disease or chronic infections of the gum tissue actually have overrepresentation of harmful organisms and underrepresentation of beneficial microbes, which correlates strongly with disease severity and inflammatory marker production. So this is a big deal and something that I'm very interested in. So the rest of the talk is just talking about how you guys can help your clients' microbiomes become healthier. And I know it's a bit abstract and difficult because you can't see them. Welcome to my world for the past 17 years. But there is very well known, very well characterized data sets that we can lean on to show outcome. And so I know that antiseptics and antibiotics are the current standard of care in veterinary medicine to tackle a lot of microbial overgrowths. both narrow and broad spectrum, realize that they do have their own chemistries. So those chemistries then bias who survives and who dies that insult. On the right-hand side of this slide is actually a staff that were able to survive exposure to the highest concentration of broad spectrum antibiotic. And you can see quite many microbes are still surviving on that were isolated from a dog treated for the appropriate amount of time on an antibiotic. And those microbes are now resistant. And so then they will take over and establish themselves on the real estate that was freed up by that antibiotic, and that animal will now have an overgrowth of this drug-resistant species. So your treatment choice can actually bias outcomes in the microbiome. It can actually lead to also more dysbiosis and comorbidities. What we've seen in meta-analyses is that treatment with antibiotic exposure 2 to 3 times tends to correlate strongly with chronic inflammatory diseases that pop up, after year 5 or 6 of a dog or cat's life, which is actually a pretty scary thing to think about. More work needs to be done to really flesh that out, but it is a trend, and a trend we need to pay attention to, and that begs you guys have heard this before, but this antibiotic stewardship, but I'm also going to say antiseptic stewardship is also as important when it comes to thinking about the microbiome and the role of the microbiome in the health of that organism. Because if you eradicate the beneficial microbes in a microbiome, it sure does make that animal's life a little bit harder. And you might predispose them to getting more inflammatory diseases later on, data shows. So one of the alternatives that are really coming on the market are called prebiotics. So those are actually compounds or nutrients in foods that have a direct effect on a microbiome, not so much the host animal. So they've been studied very well over the past 30 years, I would say, more on the dietary feed side of the equation. And now they're being investigated more thoroughly from a therapeutic side. In the human market, there are functional foods that are starting to come out, and nutraceuticals already out in the market. I've seen in several of your vet clinics, your clinics, that you offer some of those as well, which is great. So the data on prebiotics, like I said, are still emerging, but they've been shown to selectively have the ability to grow or activate certain beneficial microbes in an existing biofilm to empower them to grow. And this can dramatically alter the composition of the microbiome, honestly, as fast, if not faster, than antibiotics. So instead of an inhibitory sledgehammer you're taking to that microbiome, you're sending out these signals, these feeding signals to these microbes, and that actually stimulates them to respond much faster. So they can start to eat within about 5 minutes and churn through these signaling prebiotic molecules in quick time. Like, seriously, I can't measure them fast enough. There's no known resistance that's been identified or demonstrated, which is a great thing. And an example of that is Protectin 42 for canine dental health. And what it does is nutrients to actually simultaneously block subgingival bacterial acid production, but also activate protein catabolism pathways in the bacteria. And the result is a change in the metabolism of the biofilm, which correlates with an increased biodiversity. So you're expanding the diversity of that oral biofilm in the mouth to reduce inflammation and downstream disease processes, which is a pretty new, novel approach. Probiotics have been on the market longer than prebiotics. Those are usually living organisms that are either dried down or refrigerated. And you administer them normally orally. We're seeing some now topical products on the market, and they tend to provide very transient benefits. And the reason is that they're always subject to colonization resistance, which means that that animal is already coated with its microbiome, right? Which means you're introducing probiotics and they're going to have a transient effect. They can't attach to the GI tract of that cat or dog because it's already chock full of microbiome. So it passes through. But while it passes through, it's been shown that they can statistically significantly impact the health outcomes or certain health measures in that animal. An example is E. coli strain Nissel, which was discovered in 1917. That's actually administered to animals to stop pathogen growth in the gut. And it also has been characterized to stimulate anti-inflammatory pathways that kind of quiet down gut inflammation. And it helps to restore and facilitate gap junction function. So it restores leaky gut. The great thing about it, probiotics, are that it's self-limiting. So as soon as that organism passes through the GI tract, those benefits go away. A newer and more emerging technology, which is really exciting, are called live biotherapeutic products. So these are living organisms like probiotics, but they have been genetically designed for a specific disease state. And obviously, because it's a therapeutic, living biological therapeutic, it's going to take a lot longer to get to market. It has to go through all the regulatory hurdles, the clinical, to show safety and efficacy. But there are actually several in the pipeline that one or several should make it to market in the next 5 or 6 years. NCN is actually an E. coli that's been engineered to make a phosphatidylethanolamine product that has been shown to ameliorate the effects of insulin resistance and adipose tissue growth in obese animals that are fed a high-fat diet. And so it's obvious implications in production animals, and also in human healthcare as well. So the take-home learnings, I think, hopefully you've garnered from this talk, is that, the microbiome is a really important thing, and it's been understudied, undervalued, and you'll be hearing about it more and more as time goes on. But it's been shown as becoming a major part, a driver in animal health and disease. Obviously, it's still work in progress. I mean, I'm doing my best to understand as much as possible. More and more data is on the market, is in journals all the time, so it's kind of hard to keep up these days. But I just want to plant a seed in you to be thoughtful about the role it might play. Particularly when you're assessing a client, or when you're having to make selections or choices on what kinds of intervention to select. Because I think that the microbiome has been involved in some adverse events that definitely is shown on the human side in pharmaceuticals. But take that in consideration when you're making your choices, because it's definitely a driver of successful outcomes. And the really cool thing is there's, folks like me in the market. We're building options for you so that you can make the best choice possible to really manage your client's microbiome effectively. And, this is a brand new area of interest in veterinary medicine, and I'm learning as a microbiologist veterinarians' needs. And I really, I'm just throwing it out there. Let's keep in touch, please. Feel free to reach out to me, emily.stein@primaltx.com. Always happy to talk to really excited folks about the microbiome and share some ideas. And I just want to leave you with one last quote that I came across from a fellow micro nerd. but he reflected on it that our gut microbiome is a direct extension of the nature we touch. So everything we take in affects it. And it's kind of like the characterization of the microbiome is the aftermath and accumulation of every single insult or event that an animal or a human has experienced. And with that, thank you so much for your attention. And I am very happy to stay in touch. Thank you.